Hello students, welcome to Sir classes. Today our topic is cell and development, and in this video I'm going to discuss about different types of cells and their features. So now let's get started. The first one we are taking is, we know that cells are of different types and two main types are prokaryotic cells and eukaryotic cells, and at first I'm going to discuss about prokaryotic cells. So now the first one is prokaryotic cells or prokaryotes. Okay. Now these prokaryotes or prokaryotic cells are small in size and are exhibited by bacteria and cyanobacteria, that is blue-green algae. So these cells are small sized and are exhibited by bacteria and cyanobacteria which is blue-green algae. Now they have some characteristic features, and I'm going to discuss this through some points. So the features of prokaryotes. Now the first one is, the cell wall is made of murine derived from amino acids. Amino acids means glutamic acid, alanine, and glycine and lysine. So in this type of cells, prokaryotic cells, the cell wall is made of murine derived from amino acids. That means glutamic acid, alanine, glycine, and lysine. Now sometimes instead of lysine, similar substance like instead of lysine, some similar substance like N-acetyl, glucosamine, and N-acetyl muramic acids. Okay, muramic acids are present. So, the first point about this prokaryotic cells are the cell wall is made of murin derived from amino acids, and amino acids such as glutamic acid, alanine, glycine and lysine. Now, sometimes instead of this lysine, similar substances like N-acetyl glucosamine and N-acetyl muramic acid are present. Now, the next point is plasma membrane forms enfoldings called mesosomes. Plasma membrane forms enfoldings which is called mesosomes. Or chondrioids, which control DNA replication, and this controls DNA replication. So, plasma membrane forms enfoldings, and this is called mesosomes, and these mesosomes help in controlling the DNA replication, cell division, and respiration, as they store respiratory enzymes, and respirations as they store respiratory enzymes. Now, the next point is the membrane bound organelles such as mitochondria, plastids, Golgi bodies are absent. Now, in this type of cell, prokaryotic cells, the membrane bound membrane bound elements such as mitochondria, plastids, Golgi bodies, are absent. So we can't find this mitochondria, plastids and Golgi bodies, which are the membrane bound elements in prokaryotic cells. Now the next point is flagella if it is present, it means this flagella can be present or cannot be present in prokaryotic cells. So if present, lack 9 plus 2 fibrillar, okay, it was 9 plus 2 fibrillar structure but have a single fibril made up of flagellin proteins, So the flagella, if it is present in the prokaryotic cells, then lack 9 plus 2 fibrillar structure, but it have a single fibril made up of flagellin proteins. Now the next point is here in these prokaryotes, cytoplasmic streaming that is called cyclosis is absent in these prokaryotic cells. The cells divide by simple fissions. These cells divide by simple fissions. So these are the main features or characteristic features of prokaryotic cells. Now we are going to discuss about eukaryotes or eukaryotic cells. So the second one is eukaryotes. Now These eukaryotic cells contain a membrane-bound nucleus having nucleus and chromosomes and membrane-bound organelles. So, this eukaryotes or eukaryotic cells contain a membrane-bound nucleus having nucleolus and chromosomes and membrane-bound organelles and membrane-bound organelles. So, in eukaryotic cells or eukaryotes contains a membrane-bound nucleus having nucleolus and chromosomes and membrane-bound organelles. But just to remember you, in prokaryotes or prokaryotic cells, we cannot find these elements. Again, these eukaryotes are of two types. These eukaryotes, we can write it like eukaryotes are of two types. One is plant cells and the second one is animal cells. So we can find two types of eukaryotes, plant cells and animal cells. Now let's discuss about plant cells. Plant cells. A typical plant cell can be differentiated into three parts. Now, it can be differentiated into three parts. These are cell wall, again protoplasm, and vacuole. So, with these three elements, a plant cell can be formed. So, it can be differentiated into three parts. Now, let's discuss about protoplasm. The term protoplasm was first given by Purkinje. It was first given by Purkinje in 1839 for the living substance of the cell. For the living substance of the cell. In 1846, Hugo in 1846, Hugo introduced a distinction between the protoplasm and cell sap. Introduced a distinction between protoplasm and cells. Now the protoplasm is a viscous colorless fluid. A protoplasm is a colorless fluid and is the seat for all the physiological functions and is the seat for all the physiological functions. Later, in 1868, Huxley suggested that protoplasm is the physical basis of life. So, the term protoplasm was first given by Purkinje in 1839 for the living substances of the cell. In 1846, Hugo introduced a distinction between protoplasm and cells. A protoplasm is a viscous colorless fluid and it is the seat for all the physiological functions. In 1868, Huxley suggested that protoplasm is the physical basis of life. So in this video I have given the details about the different types of cells, that is prokaryotic cells, eukaryotic cells, and you can pause my video anytime and watch it, repeat it as you need. Thank you.